thanks very much to New Phytologists and the organizers for organizing this and inviting me. I'm very, very happy to be here. It's the last talk of the conference, and there's going to be some chemistry in it, so I hope that won't be too painful. But I'm very interested, and my group is very interested, in the molecules that plants make. And I think you've heard a little bit about this over the last day or two. But just as a reminder, plants make many, many molecules that have fabulously interesting and important applications. So for example, digoxin, which comes from common foxglove, has been used to treat a variety of cardiac ailments for hundreds of years. And to get these plant-derived compounds into the clinic, typically, even today, we extract them from the whole plant, or we extract advanced precursors from the plant and then do some chemistry on them. And so the hope is, and I think that many of you already know this or are actively contributing to this, is that next generation sequencing, bioinformatic approaches, and what we loosely call synthetic biology really have the power to bring the medicinal plant metabolism really into the 21st century. And this is just a slide with just 4 samplings of secondary metabolites or specialized metabolites or natural products. Those terms are used somewhat interchangeably, that are produced from plants, and they all have very important uses. The names may be recognizable to many of you. They're all used in the clinic for one reason or another. And the point that I want to make is that the biosynthetic pathways that construct these molecules have not been completely elucidated for any of these molecules. And so given the importance of these molecules and the sort of the hundreds and thousands of graduate student and postdoc hours that have been put into studying these biosynthetic pathways, I think that's really sort of a testament to how challenging understanding plant metabolism can be. So what I want to do is talk about our group's approaches and progress in studying how plants make some of these complicated small molecules, and we're interested in understanding these metabolic pathways partly from sort of a fundamental biochemistry perspective. We're very interested in how plants do complex chemistry. We're interested in understanding how these enzymes catalyze these reactions that put these molecules together, and potentially understanding how and why the plants put these enzymes together in these pathways in the way that they do. And then, of course, there's also obvious practical benefits to understanding plant metabolism. So, for example, vinblastin, which is made by Madagascar periwinkle or Catharanthus roseus. This is a potent anti-cancer drug that acts on microtubule formation. It was discovered in the middle of the 20th century. It's isolated from the leaves of Catharanthus and very low yield, hence it's expensive. We could imagine that if we knew all of the enzymes that made up vinblastin biosynthesis, we could reconstitute those enzymes in a different host organism like Nicotiana benthanyama or yeast or E. coli, and then maybe trick that organism or program that organism into making larger quantities of vinblastin. Or maybe we could even fiddle around with the biosynthetic pathways and instead of making vinblastin itself, make analogs or derivatives of vinblastin that might have new and improved biological properties. Okay, so how do we go about thinking about how a natural product or a specialized metabolite is made? And you know, a lot of times you just sort of see a bunch of structures put up on a slide representing the start to finish, and a lot of work typically went into drawing that or coming up with the rationale behind that figure. And just very quickly, I'm not going to get into it, really the background behind this slide has been a lot of labeling studies. So in other words, taking isotopically labeled precursors and feeding them into the plant and seeing which ones get incorporated into the final product and which ones don't. A lot of isolation of biosynthetic intermediates, so grinding up the plant, trying to fish out these very, very small quantities of various intermediates along the way in the pathway, and then solving their structures. And then using chemical intuition in combination with these two approaches to try to put a chemically reasonable set of steps together that would lead to the final product, utilizing the structure of the intermediates that hopefully you've been able to elucidate. Okay, so how is vinblastin made? Well, you can see it's made up of two sections, both of which have an indole, or in this case, something that looks something like it was derived from an indole. And if you see an indole in a specialized metabolite, it probably means that the amino acid tryptophan was involved. And in this case, the plant actually decarboxylates tryptophan to make this tryptamine here, so just lacking that CO2 there. You can also see there's a whole bunch of other carbons on both of these monomers as well, which I've highlighted in red. And if you count the carbons, and again, having a little bit of chemical intuition and using some labeling studies, that tells you that these red bonds here are derived from a monoterpene. And by isolating some biosynthetic intermediates, people were able to determine that actually the monoterpene involved in vinblastin biosynthesis is this very crazy looking monoterpene, which I'll talk more about, which is called cyclogenin. And if you put tryptamine and cyclogenin together, it's very easy to see that how they come together to form this other isolated biosynthetic intermediate, which is called strictocidin, which can then go on to rearrange to form these more derivatized compounds here, catharanthine and vindoline. And it's, again, fairly straightforward to map catharanthine onto one half of the vinblastin molecule and vindoline onto the other half of the vinblastin molecule. And presumably the plant dimerizes these two advanced biosynthetic intermediates together to give the final product. And in fact, in the commercial production of vinblastin, typically, vinblastin itself is not isolated from the plant, but catharanthine and vindoline are isolated, and they can be chemically dimerized together in a fairly straightforward chemical synthesis. So in terms of the enzymes that are used to construct these, or to actually catalyze these chemical conversions, we only know, or at the time that we started this, we really only knew a fraction of them. So we know there's a tryptophan decarboxylase, which does this conversion of tryptophan to tryptamine. We really only knew 1 out of 3 or 4 of the enzymes required to convert the starting terpene, geraniol, into this intermediate of cyclogenin, which is called nepentolactol. And cyclogenin is a type of monoterpene known as an iridoid, and nepentolactol is the common scaffold for this class of terpenes known as iridoids. And it was not at the time known how nepentolactol was synthesized from geraniol. We know the enzyme that condenses tryptamine and cyclogenin together. We know that strictocidin gets deglycosylated by a dedicated glucosidase enzyme. We know very few of the steps after strictocidin glucosidase. We do know some of the tailoring enzymes that put some of the functional groups on vindoline, but that really is about it. Okay. So we decided to focus on this part of the pathway, partly because it made sense to sort of begin at the beginning, particularly if we were interested in thinking about reconstituting this pathway in host organisms. But also we were from a chemical perspective, we were very intrigued by the chemistry involving the conversion of this fairly simple linear molecule into this much more complicated fused ring structure here. Okay, so as I mentioned, this represents iridoid biosynthesis, and iridoids are a specialized class of monoterpenes that are derived from geraniol. And so what we knew at the time is that geraniol is the committed precursor, and this is hydroxylated by a P450 to give this dihydroxy compound. And then this hydroxy compound is oxidized by an alcohol dehydrogenase enzyme to give this dialdehyde. And it's this dialdehyde that's actually the immediate precursor for this compound nepentolactol, which, as I mentioned, is the framework of the iridoid skeleton. This is the common intermediate for all iridoid monoterpenes. And this 5-6-membered ring structure that's fused together, this is really the kind of the definitive structure of the iridoids. So nepentolactol, of course, goes on to form the downstream alkaloids in Catharanthus roseus, like vinblastin. The iridoids are also an interesting set of structures in and of themselves. The simplest iridoid that's found in nature is nepentolactone, which is found in Nepentocataria, which is the active ingredient in catnip. So I actually hadn't really realized this until we started working on this pathway, and I was very excited to be working on catnip. You never sort of expect these things when you start working on a pathway. In addition to that, slightly more important applications of these compounds. Some of them have pharmacological activity. Many of them also have important uses in agrochemistry. So these iridoids are either produced or sequestered by various insects, and these play a role in sort of signaling with insects and can be used as repellents or attractants, which is very, very important. Okay. So the other point that I want to make, and this was the other reason why we were very interested in this section of this pathway, is that this biosynthesis, this chemistry, it's very different from how normal or canonical monoterpenes are made. So normally, nature makes this precursor called geraniol pyrophosphate, and then a terpene cyclase will catalyze the ionization or the leaving of this pyrophosphate group to generate a carbocation. And then the combination of this carbocation and the double bonds in the molecule then facilitates a rearrangement process to give lots and lots of different possibilities. I've just shown limonene here as one example. Additionally, this carbocation intermediate, instead of cyclizing, can also be quenched with water, so a molecule of water can just come in and attack here, and that's actually how geraniol is synthesized. So then in the case of iridoid biosynthesis, we start off on this pathway, but geraniol is made and then gets fed into this alternative branch. All right. So how do we go about figuring out what this missing step is? And we've used what many, many other groups have used very successfully, namely transcriptomic data. And this is part of an NIH-funded program called the Medicinal Plant Genomics Consortium, and you can find all of the background as well as the raw data on this website here. So how do you go about using transcriptome data to fish for gene candidates? And I know many, many of you in the audience already know this. We use co-expression analysis. So the idea is that if you can guess what type of enzyme might be required in your chemical transformation, like for example, if you're looking for something that puts a hydroxyl group on a molecule, you might guess that a P450 is involved. That's not a bad guess. And then we make the assumption that all genes in a pathway will co-express. They'll all have similar expression profiles. So we look among our contigs that for example, would be annotated as a P450, and we select the annotated P450s that are closest expressing with respect to a known gene. So for example, we take vinblastin biosynthetic genes that have been identified earlier, and then we look to see what, for example, P450s might be expressed most similarly to those known genes. And there are some problems with this approach. For example, the contigs might be not annotated or they might be misannotated. And actually, in the case of looking for this iridoid synthase enzyme, our guess of what type of enzyme might be wrong. So this is particularly true for the most interesting and unprecedented type of chemistry. You might not know what type of enzyme actually catalyzes the reaction, and so it's difficult to make an initial guess based on the protein family. So really a better approach is to try to do a database-wide co-expression analysis. And to try to make this problem a little bit simpler, we try to think of clever ways to do, or straightforward ways to do an initial filtering of our contigs. So for example, in the case of vinblastin biosynthesis, we know that vinblastin and all of its precursors are synthesized in leaf. So we were able to eliminate many, many transcripts based on a filtering process where the transcripts had to have some baseline level of expression in our leaf tissues. And then we also were able to obtain RNA-seq data for seedlings that had been subjected to methyljasminate treatment. And it was well studied that methyljasminate will upregulate production of vinblastin and all of its precursors. So we set in a filtering where we had to see higher expression of of contigs in these methyl jasmonate treated seedlings. And so what we could see when we looked at our transcriptome data or RNA-seq data is that we could see the genes from the early part of the vinblastine biosynthetic pathway that includes this iridoid section of the pathway are actually arranged in a cluster, essentially, of, and in this case, of about 100 or so transcripts. So this was really fantastic news, because trying to look for an unknown enzyme in a group of 100 is a lot easier than looking for an unknown enzyme in the 35,000 or so genes that are expressed in Catharanthus roseus. And so we specifically focused on looking for genes that co-expressed with this P450 that puts the hydroxyl group in this position here, G10H. And so what we could do is, we could just sort of start at the beginning and go through 100 or so genes and look to, that were most similarly co-expressed with G10H and look and see which one did the job. But it's actually nicer if you can narrow that down a little bit further. And this is where some chemical intuition really helped streamline this process. And we were very, very fortunate because Meinhard-Zenk's group back in the 1980s actually looked into this problem a little bit. And Zenk was able to take crude plant extracts and incubate them with this substrate. And what he found was that to get maximal activity, you needed an NADPH cofactor. So that was a big clue to us that this enzyme that catalyzes iridoid biosynthesis is an enzyme that requires an NADPH cofactor. And when we go and we look at our list of genes that share expression similarity to G10H, one of the first hits that we saw was a contig that was annotated as progesterone 5-beta reductase. And we know that this is a gene that requires NADPH. So the progesterone 5-beta reductases are found very widely throughout the plant kingdom. Biochemical studies have shown that they have broad substrate specificity. And the endogenous functions, the native functions of these enzymes in plants are just not that clear. In many cases, they're believed to be involved in triterpenoid biosynthesis, cardinalide biosynthesis, but they're also found in plants that don't make these compounds. So it's a little bit of a mystery what these enzymes are doing much of the time. But in terms of the chemistry, we know that progesterone beta reductases, they're reductants, and they use an NADPH cofactor. So NADPH basically just donates H-. This is hydrogen with an electron pair on it. And this H- is the reductant. It goes in and it attacks this double bond here. It does this sort of rearrangement to form what's called an enol. So basically, you're just moving the electrons from this double bond to here, and then generating a carbon-oxygen single bond. And then this can spontaneously rearrange to form this. So basically, you're just doing a net reduction of this double bond. Now, progesterone looks nothing like this dialdehyde substrate that this putative iridoid synthase is supposed to use. But actually, if you kind of line it up with progesterone beta reductase, you can kind of see that sort of key functional group, this double bond and this carbonyl, it's the same in both molecules. And so we could propose, using this mechanism that Zenk had written out in 1987, that the H- from the NADPH cofactor could attack here. That would enable formation of this 5-membered ring, and then we could actually get a spontaneous formation of the second ring just by attack of this alcohol onto this aldehyde here. So we thought that this progesterone reductase was actually a good candidate. And so we could clone it out, we could express it in E. coli, it expresses pretty well. We can get the enzymatic product, and we can actually characterize it by NMR, and it's identical to an authentic standard. And just sort of fun trivia fact, how did we get the authentic standard? You can actually do a reduction of nepentolactone, that active ingredient in catnip, to get this compound. And you can go on the web and you can actually buy like liters of nepentolactone. And so Nat Sheridan was distilling the essential oil of catnip and purifying nepentolactone and then reducing it. And I would bring things home from the lab, and the cat that I had at home just went berserk. Anyway, so it's fun working with these biologically active compounds. And then we can do in vitro steady-state kinetics and ensure that this is actually performing the reaction with sort of appropriate kinetic parameters. We're still working on the mechanism, the details of the mechanism of this enzyme. I'll just note that there's a key mutation compared to progesterone beta reductase in what we believe is the active site, a tyrosine to a histidine. We've made mutants of this. This doesn't really switch the activity. It doesn't convert, a progesterone beta reductase into an iridoid synthase. So there's more to the story that's remaining to be discovered, but we're in the process of trying to get some structural data for iridoid synthase, and I hope this is going to shed some light on really how this enzyme works and also maybe how it evolved from this family of progesterone beta reductases. We can also look at the function of this enzyme in vivo. So a former postdoc in the lab worked out how to do VIGS, virus-induced gene silencing, in Catharanthus roseus. And so we can silence this cyclizing enzyme and look to see a decrease in the formation of downstream alkaloid products. So that really kind of suggests that this enzyme is actually functioning in vivo. So to just think a little bit about how this enzyme works, I sort of drew the mechanism or the arrows showing the movement of the bond formation like Zenk did, where everything sort of all happened at once. And if you take a look at the stereochemistry, so whether these bonds are pointing up or down, as Nat did, this is basically impossible. There's sort of no room for this to happen. Basically just means that this cofactor sort of has to be in the middle of everything, and it's just not chemically reasonable. So we went back to the sort of the way that the progesterone beta reductase happens, and as I mentioned, it proceeds through this initial reduction here to form what I call this enol here that then spontaneously sort of rearranges. And so we think that's basically how this iridoid cyclase works, that first there's an initial reduction reaction and then cyclization happens. And to kind of show this, we looked at the substrate specificity of a whole bunch of different substrate analogs that were capable of being reduced. So in other words, they had a double bond here, but they couldn't cyclize because they were sort of missing some key component of the other end of the molecule. And what we can see is for example, we can see conversion of this substrate here. It can get reduced without being cyclized. And so what we believe is that we get what's called an enone reduction. So all that means is that we're reducing this double bond here to form that moiety that's called an enol, that exactly like what happens in progesterone beta reductase. And then once we get this so-called enol, then that's set up to nicely rearrange to form first the 5-membered ring and then the 6-membered ring by forming this bond here. And so again, we were very excited about this because really this is kind of a different way of making terpenes. So as I mentioned, canonical terpene cyclases proceed via this cationic intermediate that results from the ionization of this pyrophosphate group, followed by sort of just rearrangement and reaction of the double bonds with that cation, whereas this cyclase utilizes a reduction reaction followed by a subsequent cyclization. We're very interested in exploring how we can sort of combine the ability to do reduction with these types of substrates to make new and interesting structures, either for just chemical purposes or biocatalysis purposes. So can we generate new and interesting biocatalysts that make complicated molecules that might be hard to make from a synthetic chemistry perspective? And then also, again, from kind of an evolutionary perspective, can we look and see what other enzymes are found in plants or other organisms? And are there any similar types of reduction cyclizations that are also found in nature? And just as kind of a proof of concept, what we did was we took a reductase that is actually, it's involved in ergot alkaloid biosynthesis in a fungus. And we use this reductase because we had it in the lab, because a graduate student had been working on this biochemical transformation. And so we took our substrate that Catharanthus uses for nepentolactol formation, and we incubated it with this fungal enzyme, which is called EASE-A. And we saw, not unsurprisingly, just the reduced product. So our EASE-A just simply reduced one of the double bonds and just kind of left it at that. But we also saw a completely, this not unrespectable ratio, a completely different type of cyclase structure. So this is, and then we could show that a little bit more rigorously by characterizing the structure by NMR. So I mean, this really suggests that there's some plasticity, that if we can take several different families of reductases, we might be able to those substrates can fold up in various different ways in the active sites of these different reductases to form a variety of different structures. And so we're currently in the process of looking at lots and lots of different reductases from plants and other organisms, and also making analogs of 10-oxogeraniol that are shorter or longer or have different numbers of methyl groups on them, and trying to see sort of what different types of compounds we can make using this enzymatic reaction. So that's kind of a story about how we can use transcriptomic data to sort of find the missing pieces in these complicated natural product biosynthetic pathways. And I mentioned in passing that while one application of this work might be to make a heterologous organism that makes lots and lots of emblastin, what if we could fiddle around with the pathways to make compounds that had certain changes or certain derivatizations that might really improve or enhance the biological activity of these compounds? Now, it's difficult to reconstitute vinblastin in a heterologous organism because even though we're making progress in identifying all the enzymes, we're still not there yet. That's our goal. That's the goal of a number of groups, and I suspect it will happen relatively quickly. But for now, if we want to play around with these biosynthetic pathways, what we need to do is we need to actually do it in Catharanthus roseus itself. So using a variety of different techniques, such as feeding experiments and others, we could determine that the biosynthetic enzymes of vinblastin biosynthesis are actually relatively plastic and will accept a variety of different substrate analogs to go on and make alkaloid derivatives. And in one example, what we were able to do was we were able to take what are called tailoring enzymes from bacterial biosynthetic pathways. And the two examples I'm showing here are RebH and PeerH. And these are halogenases, and what they do is they put chlorines on tryptophans. So these are used in bacterial antibiotic synthesis, and these antibiotics that are made by these bacteria happen to have a chlorine on an indole, and these are the enzymes that are actually doing that job. And so what we had imagined was that we could take either RebH or PeerH and transform them into Catharanthus roseus. And these enzymes would actually encounter tryptophan in Catharanthus roseus, and then the native or the endogenous tryptophan decarboxylase that's found in Catharanthus roseus, we hoped, would be able to decarboxylate these chlorinated versions of tryptophan. And in fact, this was true. We actually cloned out and overexpressed tryptophan decarboxylase and did some biochemical analysis. And we were able to show that the Catharanthus tryptophan decarboxylase could, in fact, accept these chlorinated tryptophans, though at significantly decreased efficiency compared to the native tryptophan substrate. But nevertheless, we thought that it would be, based on these kinetic parameters, we thought it would be reasonable to expect that Catharanthus roseus would be able to generate these chlorinated tryptamine analogs in situ, and these are the direct precursors for vinblastin, as well as a number of other biosynthetically related alkaloids that are also derived from tryptamine. And so sure enough, we were able to do this. We were able to transform RebH and PeerH into tissue culture, hairy root tissue culture of Catharanthus roseus. And then we could purify various chlorinated natural products that resulted from this experiment. And this is just one example here. We can actually do this a little bit in a little bit more sophisticated manner. So it's actually, I don't think it's great to make a chlorinated amino acid in the plant. This amino acid is not just being used in alkaloid biosynthesis or natural product biosynthesis, it's being used for all sorts of other things as well. So it would be nice to, if we're going to make an analog, it'd be nice to do that, to have that analog making reaction actually a little bit later on in the pathway when the pathway is actually fully committed to natural product biosynthesis. And so we know that we won't be interfering with any other pathways. And so we had the idea that instead of chlorinating tryptophan, what if we just directly chlorinated tryptamine? And so if you happen to know the structure of the active site of the chlorinating enzyme, and we did, because Kathy Drennan and Chris Walsh had crystallized RebH, we can look at the active site structure and try to make some changes to it. We did this in what we call a semi-rational way. It's not completely rational because we never honestly completely know what we're doing when we try to engineer the substrate specificity or the product specificity of an enzyme. But it's a little bit tricky to do true directed evolution with these types of reactions. We just really didn't have a good selection or a screen to enable us to go through hundreds of thousands of different mutants. So we made about 50 or so mutants sort of in the active site region. And the graduate student, Wes, just happened upon one that really was successful. He noted that this tyrosine residue seemed to interact with the carbonyl of tryptophan, and he rationalized that if we made this tyrosine, or if we mutated it into a bigger amino acid, this would disrupt the binding of tryptophan because there would be no more room for the carboxyl group to bind, but maybe it would allow binding of tryptamine. And this was, in fact, the case. So Wes could show that we saw he could transform this into Catharanthus roseus hairy roots. And when he looked for production of chlorinated tryptophan, he saw absolutely nothing. He also saw no conversion in vitro as well, but I'm not showing the biochemistry here. And then we could also see production of this compound that we saw in the previous experiment as well. So again, if we sort of know something about the pathway and we know something about the structure and the mechanism of these enzymes, we can sort of refine and make this even more efficient. And I just want to highlight, so people ask, why chlorines? What's so great about chlorines? And a lot of drugs do have halogens such as fluorine, chlorine, and bromine on them. They're good hydrogen bonding acceptors. They have some very nice pharmacological properties. So it's a sort of a good group that medicinal chemists like to add on to natural product scaffolds to see how they modulate the activity or the pharmacokinetics of a molecule. But there's another reason why chlorines are interesting, and that's because they actually serve as a handle for further derivatization. So sort of inspired by the work of Rebecca Goss, who's now at St. Andrews, who did this in a bacterial biosynthetic pathway, we did what's called a cross-coupling reaction. So if we have a molecule with a chlorine on it, we can use a catalyst under relatively mild conditions and substitute this chlorine with a variety of different organic groups, as I'm showing here. And the really nice thing about this is we can take our transformed hairy root plant tissue, we can grind it up in methanol and water, we can add in the catalyst and heat it up to 100 degrees, and then we can actually get pretty good yields of the corresponding derivatized compound. So this is a nice way to sort of further elaborate on the structure of these engineered molecules. So, and again, as I mentioned, it's a very exciting prospect to think about sort of combining these ideas with our ability now to identify the pathway enzymes in a relatively more streamlined manner. And maybe we may be able to start thinking about heterologously expressing these enzymes in a host, a heterologous host, to make these types of analogs more efficiently and more rapidly. So with that, I really want to thank the people who did all of the work. This is the current group, present and past, and these agencies for funding. And again, thank you very much to the organizers for organizing this conference. And if there are any questions, I'd be happy to try to answer them.